I do know that the movies are really wrong because they show you taking like this teeny tiny cyanide capsule and actually like to kill yourself with cyanide, if it's not super concentrated, you need like a urinal cake size thing. So you literally have to be like munching on this thing as like the cops are breaking down your door. It's not really the small capsule, but also carbon monoxide does something similar. Okay? It blocks the electron transport chain. So if somebody sits in their garage with their car on, that's why we eventually perceive it as quote-unquote death. Because part of it is, yeah, we're depriving ourselves of oxygen, but also that carbon monoxide disrupts this electron transport chain. There are other toxins that also can make the membrane of the mitochondria become leaky, so that the protons can just diffuse back through the membrane instead of having to go through ATP synthase.
So anything that involves energy is not breathing.
Exactly. Basically, anything that involves energy, you're going to get those endergonic reactions that no longer can have the energy, or no longer have the energy they need to proceed, and so they're not going to proceed. Yes?
Is there some sort of like antidote or something that would disable the cyanide energy system once you had adjusted it?
That I don't actually know. Typically with something like that, it's pretty difficult to diffuse it because in theory what you're going to have happen is that antidote is going to have to actually permeate the mitochondria and bind to the cyanide there, and then those two molecules bound together are going to have to get back out of the mitochondria. And that usually doesn't happen that way. So we do have some ATP produced. Well, what's going to most likely happen initially is if the mitochondria aren't making ATP, you're probably going to perform glycolysis followed by lactic acid fermentation, which is an anaerobic process. We're going to talk about fermentation here in just a second. That's going to make some ATP, but remember again, where does the majority of your ATP come from? Oxidative phosphorylation. If you can't perform oxidative phosphorylation, your body's going to use up any excess ATP it had. It's going to perform glycolysis, fermentation, and potentially to a degree the citric acid cycle to make more ATP, but you're not going to be able to make enough, most likely, to provide your body with the energy it needs. Okay? All right. So, typically, when we're talking about aerobic respiration, or in some instances anaerobic respiration, the way that you see most of the energy flow during this process is from glucose, eventually to NADH, to the electron transport chain, which then generates the proton gradient, which moves through ATP synthase to provide a proton motive force, which ultimately produces adenosine triphosphate. So this is the overarching idea as to how we produce ATP from a single molecule of glucose and how we produce enough to survive. Multicellular organisms, at least all of them to my knowledge, have to perform some form of cellular respiration where they utilize that proton motive force, be it anaerobic or aerobic respiration. They have to perform that to produce the large amounts of ATP that they need to survive. Smaller single-cell organisms can easily switch between a glycolysis and fermentation exclusive environment or processes to cellular respiration. Granted, there are some bacteria that can't perform aerobic processes, but if you're a larger organism, you're going to need a lot of ATP. If you're a smaller one, not so much. Yes, Rick?
I see NADH up there. What does FADH do?
FADH2 is also involved in the electron transport chain. I think the reason why they chose NADH is because we make so much more of this than we do FADH2. You can, though, if you want to, write under your FADH2 just as well. And as I told you, about 40% of the total potential energy stored in a molecule of glucose is released and utilized to make the ATP. So we're able to harness about 40% of the total energy inside a single molecule of glucose. Okay, so here's the overall view of everything we've talked about over the course of the last, what has seemed like probably a month to y'all. 2 ATPs produced during glycolysis, 2 during the citric acid cycle, and 32 to 34 produced during oxidative phosphorylation. Now, some students ask me, well, why is it 32 or 34? Like, why is it not just exactly, 32 or exactly 34? And to the best of my knowledge, this is the reason why. You're working with diffusion here. Is exactly 34 protons always going to move through there?
No.
Not necessarily. Maybe sometimes you get 32, maybe sometimes 33, maybe sometimes 34, which is why they give an estimate here. Okay? Because with these, because we're performing substrate level phosphorylation, we know we have to have X number of reactants to produce X number of products. With oxidative phosphorylation, though, it can vary. Yes, Lorraine? Are they? What are they saying? 30 to 32? So, okay. So they're actually altering even farther. So now I guess it's 30 to 32, which is an update that they've recently added. I actually know that. Yes. Okay. 30 to 32, I'll take honestly 30 to 34. So if it's anywhere in that range, I will accept it. So if it's 34 to 38 ATP, that's fine. I always learned it as 36. I never learned it as 38 at all. But then they, I guess, decided collectively that it was 32 to 34, and now they're deciding collectively that it's 30 to 32. Woohoo. Okay? Showing you that science is an ever-changing field. Okay? So 30 to 32, but for all intents and purposes, I'll accept anywhere from 34 to 38. Okay, now, let's talk about, yes?
I thought you just said that you would accept 30 to 34, but now you're saying 32 to 36 and 38. Well, it's on the clock, so that would be like
30-something.
What would I like?
30 to 30 plus something.
Because there's a discrepancy between the two books, and some people have the old version and some people have the new version, on an exam, I will accept, if they're saying 30 to 32 here, is that what they're saying? It's 30 to 32, or is it 30?
26 to 28 there.
Really?
And
26 to
total
20?
of
Okay.
30 to 32.
Okay. All right, so I will accept anywhere from 30 to 38, honestly. Since the books are different, but we have people who are working with an older edition and some working with a newer edition, I'll just accept anywhere from 30 to 38. Any questions I have that written in will be clear and distinct as to what I'm actually looking for.
You're talking about maximum per glucose at this point.
Yes,
Yeah.
exactly.
So for the
Oxidative phosphorylation.
what would you accept there?
What would I accept there?
Or will there be?
This tells me what's not on the exam.
Yeah, I understand y'all's concern, and it really kind of ticks me off that there's this discrepancy between the lecture and the, because I pulled the lecture slides from Kirsten's website, so it kind of ticks me off that they didn't update the lecture to match the book. So I'll tell you what, the lowest number y'all are seeing is 26 in the new book? Okay. So it's 26, and the highest y'all are seeing, if you have the old book, is 34. If I'm going to ask you about it, I'm going to probably give you a range of 26 to 34.
That's probably.
Yes, it will be clear cut. It will not be somewhere in the middle, or you won't have two potential answers somewhere in the middle. It will be clear and distinct as to what I'm asking. Okay, and I'll make sure that that matches. Sorry about that discrepancy. I was completely unaware that they had made that change because they didn't update it in my lectures. They told me, hey, we have a new edition of the book. Don't worry, nothing's changed. So it's nice to know something crucial like that has. Any other questions, concerns? Okay. Moving right along. Let's talk about anaerobic respiration and fermentation. Two separate processes. Okay? So just so you know, the majority of cellular respiration does require oxygen, but there are select types where something can be substituted in for oxygen. Okay? So sometimes the final electron acceptor will be something other than oxygen. This is if we're actually utilizing the mitochondria and the citric acid cycle. If we're only staying out inside the cytoplasm, we're going to perform glycolysis, and we're most likely going to follow that with some form of fermentation. And so if we don't have oxygen, we're either going to perform fermentation or we're going to perform anaerobic respiration. So again, anaerobic respiration still utilizes the electron transport chain, but it winds up producing, or I guess donating the final electrons to something other than oxygen. Usually something like sulfate, which is commonly used. Fermentation instead never enters the mitochondria. Instead, what we're going to do in fermentation is we're actually going to use NAD plus and NADH as what's known as an electron sink. In doing this, we're going to transfer electrons from molecule to molecule. Okay? And as we do that, we're going to endlessly fuel glycolysis. And I'll show you that in just a second. Okay? So, NAD plus and NADH are reused for glycolysis. Now, depending upon what kind of organism you are, you're going to perform a different type of fermentation. Yeast perform what's known as alcohol fermentation, which means at the very end of fermentation, the final product that is produced is alcohol. We do not do that, thankfully. Okay? Instead, human beings perform what's known as lactic acid fermentation, which means at the very end of fermentation in humans, lactate is produced. So let me go ahead and show you first and foremost, this is alcohol fermentation. We perform glycolysis the same way that we've done before. Okay, so just as we have previously, we perform glycolysis where we have 2 ADPs and 2 phosphates forming 2 ATPs. And then, through that, again, we're going to produce pyruvate. We're going to remove 2 carbon dioxides from that pyruvate, and we're eventually going to convert that pyruvate into ethanol through, again, another reduction oxidation reaction. So in this particular exercise, what's happening is NAD plus is becoming reduced, as we've seen previously in glycolysis. It's going to be reduced to NADH plus those protons. And that NADH and those protons are going to be used to reduce acetaldehyde to ethanol. We produce alcohol, and we again reproduce the NAD plus by performing that redox reaction. Okay? So, pretty straightforward. You already know glycolysis. You know what's produced at the end of glycolysis. Now all we're doing is we're removing carbon dioxides and using the electrons and hydrogens associated with those that we made when we made NADH to reduce these to ethanol. Questions?
You say we don't do that.
Okay, say we don't do that?
You're saying that we don't do that because I had heard.
We don't do this, which is good.
Well, I had heard that the human body makes a little bit of alcohol, but I don't know if that's true or not.
Well, the human body has the potential to make certain types of alcohol, but it probably doesn't make a lot during fermentation. Otherwise, relay races would be really interesting. Okay? Your runners would probably start stumbling around a certain mile marker. Actually, the more likely thing that would happen is I imagine your muscles and the rest of your body will probably just literally explode. We instead perform lactic acid fermentation. It's going to look almost exactly identical to alcohol fermentation, but instead of producing ethanol, we're actually going to produce lactic acid. Okay? So again, we perform glycolysis. We generate those NADH molecules and those protons. We're going to take pyruvate and we're going to reduce that pyruvate down to lactate. Okay? What's that?
Would alcohol fermentation be like potatoes used?
Typically, alcohol fermentation is the organism used to make or to have alcohol fermentation is usually yeast. There could be some microbes associated with it as well. But, right, if you let potatoes sit, or I guess you ferment them, and that's why it's called fermenting to make. Are you talking about to make certain types of alcohol?
Yeah. Well,
yes. Okay. Anytime something like that is being generated, it's typically done through a means of